Today, I'd like to talk about the biology of ageing, why it is an important topic, and what we might be able to do to minimise its negative health impacts. Now, we all have ideas on what ageing is, but to unpack it fully, we have to ask this question at different levels. How do populations age? From this, we can extract information and statistics like median lifespan, maximum lifespan, quartiles, and mortality rates. But we can also operate at the level of individuals. For example, how has some measure of performance changed with age? Questions of cognition, memory, mobility, or aerobic capacity. We could also move to the level of organs and tissues within an individual, where we could explore age-related loss of specific organ activity or signs of inflammation, such as fibrosis, etc. At even finer levels, we could look at changes to cells. For example, their morphology or evidence of damage to proteins, genetic mutations, etc. And in reality, this narrowing down could continue further. However, let us begin operating up here today at the population level and define ageing as an increase in the probability of death with increasing chronological age. In addition, ageing is also accompanied by a loss of vitality and fertility and increased susceptibility to disease as an organism traverses its lifespan. So why is it important to understand ageing? Well, ageing is the biggest risk factor for many diseases. The incidence of dementia and neurodegeneration increase exponentially with age. An estimate from a recent WHO study are that worldwide, there will be 10 million new cases of dementia this year. This means one new case approximately every 3 seconds. The number of people living with dementia worldwide is estimated at 55 million, projected to reach 78 million by 2030, and just under 140 million by 2050. However, if we could delay the onset of disease, like Alzheimer's disease, by 5 years, this would lower the dementia prevalence by 40%. This would result in a huge benefit to individuals, their families and carers, and society as a whole. So my big picture research goals are to blunt the future scale of this dementia epidemic. And to do this, we need to understand why ageing increases the risk of specific diseases, like Alzheimer's disease, and ultimately to improve the health of our aged population. Although this is not my speciality, demographics tell us that ageing is complex and not constant. And here's a really interesting study showing US data on age-specific death rate per 100,000 individuals on a log-log scale. The columns plot all causes of death, and then divided into death by heart disease and death by cancer. And the second row shows the acceleration of this death rate, i.e. the slope of the curve across age. And what it shows is that there's a steady rise in death rate as it accelerates through adult life. However, at late life, it stops increasing and perhaps even starts to slow and decrease. So what is special about these relative few who are aging so successfully in late life? This slowing down of mortality rate is seen in other animals too. Here's a very interesting study from 1982 looking at the aging in medflies, using some very impressive numbers, more than 1.2 million flies per experiment. And shown here, clearly once again, is this deceleration of age-related risk of death in older animals. Again, this is suggesting that some animals are aging much more successfully than others. And the obvious question is, why does this occur? And understanding the answer to this question allow us to help more people age more successfully. So how do we age? Well, it's complicated. Over the last few decades, science has provided great insight into the complicated process of aging. Although we know it's complex, we have an increasing understanding of some of the overarching processes. Attempts to simplify this model agree that growth and reproduction require energy, and normal metabolism that provides this is also complicated, but mistakes are made and need to be constantly corrected or repaired. The idea that cellular maintenance becomes impaired or is insufficient or perhaps even deleterious in late life is a common model across most theories of aging. However, what is very clear is that aging is associated with oxidative damage to cells proteins, and DNA. Oxidative stress describes an imbalance between the production and accumulation of byproducts of normal oxygen metabolism, and these are called reactive oxygen species. Reactive oxygen species are free radicals that contain oxygen, and these react with other molecules inside our cells and tissues. Free radicals are molecules that have at least one unpaired valence electron, and this makes them highly reactive and typically short-lived. In high quantities or in the wrong place or at the wrong time, these can be damaging to other parts of our cells because these molecules steal electrons from other atoms. For proteins, this may change their structure and so stop them from working correctly or make them act inappropriately. We also know that certain environmental factors such as UV light, ionizing radiation, and pollution can also affect reactive oxygen species accumulation through the production of free radicals. However, as I alluded to earlier, most reactive oxygen species are in fact produced by our own cells, by our mitochondria. It is very important to note that reactive oxygen species also play an important role in processes such as immunity and wound healing. So, production of free radicals is not always a bad thing, and when balanced, they are essential for our health. However, when imbalanced, cells lose function. Cells can die or perhaps start the process of becoming cancerous. It's also well known in the biochemistry of iron that the biochemistry of iron is a major contributor to the production of free radicals via a process like the one shown here called the Fenton reaction. But let us take a step back for a moment. You probably know that the vast majority of our cells and tissues are made up of these elements shown here: hydrogen, carbon, nitrogen, and oxygen. However, biology needs other elements to sustain life. And iron is by far the most abundant transition metal found in our cells, where the term transition metal means any metal able to make chemical bonds with other atoms. Iron is essential for life. Every organism requires iron, with the exception of a few rare bacteria. Without it, cells cannot perform essential activities like energy production, DNA replication, or detoxification of metabolites. The ability of iron to spontaneously redox cycle between ferric, Fe3+, and ferrous, Fe2+, is what makes it so important to cells, but it also makes it a potential major driver of oxidative damage. And so there's an optimal range for iron in health. Iron is vital for normal brain development and function, so too little iron can cause a host of problems, such as iron deficiency and anemia, both very serious health conditions. If unregulated, iron can be highly damaging, so having too much iron is also a problem. Excess iron can lead to uncontrolled production of free radicals, causing damage to cells, particularly lipid membranes, which is a subject I'll return to later. So consequently, there are a range of storage and chaperone pathways to ensure our iron is managed correctly. So what chemical changes occur in the brain that increase the risk for neurodegeneration? Is there an oxidative damage link? And could iron be the problem? Well, it's been established for many decades that animals accumulate iron as they age. Seen here is a study using Drosophila melanogaster, a classic genetic organism, also called the vinegar fly. And these have been reared at 25 degrees. This paper was published in 1985. And what we can see here is clearly an increase in iron as these animals move across their lifespan, which typically runs for approximately 80 days maximum. And these different shaped icons here are representative data from independent experiments. So total amount of iron shown on this axis and the age of these animals shown on this axis. Importantly, for diseases of the brain, this relationship is also seen in human populations. The aging human brain accumulates iron. Seen here is a post-mortem study published in 1958, looking at the amount of iron found in brain tissue. And a more recent report on living tissue by quantitative susceptibility mapping, or QSM, a non-invasive magnetic resonance imaging technique, also shows this increase in brain iron across adult age. This accumulation has also been robustly replicated by numerous animal studies by researchers here at the Florey. Although not necessarily the primary focus of each of these studies, Each time these experiments see that normal laboratory mice, as they age, they accumulate iron. Examples found here, again, increasing brain iron here, and again. Overall, this age-dependent accumulation of iron is consistently observed by the wider scientific community. This accumulation of iron is not just restricted to the brain. For example, mice accumulate iron as they age in skeletal muscle, liver, fat, and kidneys. And as a prequel to later slides, these tissues also show elevated evidence of damaged lipids. So, why does iron accumulate? We know that iron is critical for growth and development, and consequently there are mechanisms to regulate its uptake from dietary sources. However, animals lack a regulated mechanism for iron excretion. Consequently, iron accumulates in late life and may contribute to age-related pathologies, cell death, and reduced health span. And this has led us to ask, is needless iron accumulation a universal feature of aging? So what would be the consequence of excess iron? Although the connection between iron and lipid damage has been known for many years, it was not until 2012 that an iron-dependent specific cell death process called ferroptosis was identified. Ferroptosis may provide the link between iron and features of neurodegeneration and aging. In essence, iron initiates damage to lipid components of our cell membranes that then cause a further cascade of damage that ultimately triggers that cell to die. It's important to note that we cannot simply stop production of all reactive oxygen species. As I said earlier, some activity is needed and vital for health. For example, cell signaling, wound healing, and immunity, to name a few. So the questions that we're asking in my lab: how does iron homeostasis, the management of iron, fail with age? Do iron and ferroptosis contribute to aging? And to answer this question, we use this model organism here, Caenorhabditis elegans. This is a microscopic free-living nematode that is optically transparent, has a simple body plan, and has characteristics that make it ideal as a system for genetic studies. We use C. elegans to explore these questions because many characteristics that are central to human biology exist in C. elegans. Embryogenesis, development, nerve function, behaviour, and aging, and how they are determined by genes and the environment are all fundamental processes and all can be modelled in C. elegans. Now, just like us, these animals are conceived as a single cell, which then develops and grows into an adult. It has a nervous system with a brain and neuropil-like structure in the head, exhibits behaviour and is even capable of rudimentary learning. These animals mate, and after reproduction, they gradually age, lose vigour, and finally die. These animals were first introduced into genetic laboratories by Sidney Brenner at Cambridge. He would later go on to share the award of the Nobel Prize in Physiology or Medicine in 2002 for pioneering discoveries in the field of programmed cell death. Brenner believes that biological research required a model system that could be grown in vast quantities in the lab, were cheap to maintain, and had a simple body plan, and he chose C. elegans to fill this role. And for the curious in the audience, this is what they look like underneath the microscope. Quite an attractive animal. So C. elegans has helped make some significant scientific breakthroughs. It was the first multicellular organism to have its genome fully sequenced at the end of 1998. The first multicellular organism to have its cell lineage fully determined. That is, how each cell then divides and differentiates to become an adult worm, and each adult worm has exactly 959 cells. The first animal to have an effective wiring diagram of all of its synaptic connections in its nervous system. So these animals have 302 neurons, and we know how each one is synaptically connected to each other. The fundamental and conserved process of gene expression regulation by microRNAs was discovered and characterized in C. elegans. And it was the first animal to have the transgenic expression of green fluorescent protein, a breakthrough that had completely changed the way biomedical research is performed today. So specifically, why use C. elegans to understand aging? We need to go back in time again to some pioneering work by Michael Klaas in Colorado. He demonstrated that it was possible to isolate mutants with increased lifespan. These mutations were ultimately mapped to a gene called AGE1. This gene codes a PI3 kinase. Now, the details about AGE1 and PI3 kinase are not important for today, other than to say it's involved in a cell signaling process that's very similar to our insulin signaling. So this is a major breakthrough in aging research. It's the first demonstration that single genes could have a profound effect on an animal's lifespan and aging. These studies led to our current understanding that conserved signaling processes can alter a way an animal ages. Perhaps the biggest advantage of using these animals is their relative short lifespan. With the median lifespan of just 10 days, where median is the age at which half of the initial population is still alive, shown here. The second compelling advantage of C. elegans is the existence of these long-lived mutants. An example is shown here. Now, the genetic details are not important other than to say that this mutation in a gene called DAF2 encodes an insulin-like receptor in C. elegans. If we mutate this single gene in these animals, these animals live twice as long. We can see fraction of the population is shown on the y-axis, and age in the adult age is shown in days on the x-axis. So we can take this kind of data and we can transform it to look at mortality rate, which is the slope of this curve here. Here are the wild-type cohorts, and these are these long-lived insulin signaling mutants. And it's clear to see that the slope of these curves is different, and that tells us that these animals are aging more slowly. These mutants age more slowly than their wild-type counterparts, and this provides us with a unique tool to better understand the basic processes of aging. And for those who are curious, manipulation or genetic variation in insulin-like signaling also has conserved effects in mammalian aging, including humans. So what we can discover in C. elegans may very well relate to what is occurring in humans. So one common question that I get asked is, why don't these long-lived C. elegans mutants exist naturally, since they appear in some senses superior? Well, this is some work I published a number of years ago with Nicole Jenkins at the Florey. And if pitted against their wild type or normal counterpart in a mixed culture, these long-lived mutants rapidly die out. We can see that here. This is just the proportion of the population that is carrying this mutation that gives it a long life. If we look at this mixed population, the long-lived mutants die out within 7 generations. If this mixed culture encounters challenges like food limitation, this extinction is even more rapid. So put simply, they don't exist in the wild because they are in fact very, very inferior. There's a trade-off between extreme longevity and early life traits. We also need to remember that the big picture here is to increase the healthy years in longevity rather than simply prolonging life and prolonging late life in ill health. From an initial characterization of mutants that alter lifespan, the words lifespan and aging have often been used interchangeably. However, lifespan is a single parameter that defines the amount of time an organism is alive. Lifespan, however, does not give any indication for how an animal is actually aging, nor does it give any detail about the health of the animal. For this, we may need to use health span as a term that describes the time an individual is active, productive, and free from age-associated disease. So health span is starting to become a major focus of aging research. From a laboratory perspective, we're looking for interventions to increase lifespan and health span. And if we look at this hypothetical survival curve, what we want is an effect that maintains good health into late life, so-called squaring of the curve, rather than extending maximum lifespan of a few individuals in relative poor health. Pardon me. So, to begin our study of aging, we examined young C. elegans at this stage of the population survival shown by the arrow, with Pearl staining, a method for looking at iron in cells and tissues. This technique derives its name from the pathologist Max Perls, who just first described this technique in 1867, and it's still a technique used by histologists today. Here, the iron in these young C. elegans has been stained a brown colour. And what we can see is defined localization of stored iron in these animals in their intestinal cells, which is effectively the equivalent of our liver. So this is what young, healthy animals look like with their storage of iron. Next, we examined animals at the median age of the population, shown here by the red arrow. In contrast, aged animals, which we've done nothing to except for allow them to grow old, have a very different story. Not only have they accumulated a lot more iron, you can see by the brown staining, this excess iron has escaped its normal confines and has started to infiltrate other tissues and cellular components like cell nuclei, shown by these arrowheads. These images really suggest these animals are in a lot of trouble. Now, Pearl staining is a wonderfully descriptive technique. However, it does not actually capture all the iron within the cells, and nor does it provide a quantitative estimate of the total iron. For that, we need more modern techniques, and so we applied micron-scale X-ray fluorescence microscopy to answer this question. And for that, we turned to the Australian synchrotron. So X-ray fluorescence microscopy is an imaging technique that uses highly focused X-ray beams that knock out inner shell electrons from atoms. And as the outer shell electrons fill that void, they release photons with a characteristic wavelength that reports on exactly the chemical nature, the identity of that element. So working in collaboration with beamline scientists at the Australian synchrotron, we established methods to image and measure iron at incredible resolution within these intact animals. A representative image is shown here, and remember these animals are only 1 millimeter in length. So shown here is an image where the general ultrastructure of the animal is shown in grey, with its head up this end and its tail at this end, and the bright ribbon of colour is used to show the levels of iron within the intestinal cells of these animals. I should also point out that iron is found in other cells, but the intestine has the greatest iron concentration. X-ray fluorescence microscopy is wonderful because it provides hyperspectral data, which is just another way of saying it captures data on a very wide range of elements simultaneously. So we also capture information about where the sulfur is located in these animals, phosphorus, calcium, manganese, zinc, etc. And in fact, in this particular experiment, we were also able to identify trace amounts of strontium, a very rare element. We've become so adept at this technique, we're able to look beyond single specimens and started to be able to collect data on populations of animals, as shown here by this particularly nice image. From this, we could now know with great certainty how much iron these animals accumulate as they age and exactly where it accumulates. When young, we see here, day 1 refers to the first day of adulthood. These animals have 60 picograms of total iron. And when they age, this increases to over 100 picograms. And just for comparison, the average human body contains 4 grams of iron, a lot more, but these animals are clearly a lot smaller. So if we return to our long-lived mutants that I introduced earlier, seen here by this red line, these mutants have a greatly extended lifespan, but they also accumulate iron much more slowly, shown here on this graph. So this is the amount of iron per worm at different age points, day 4 referring early adulthood, and day 15 when these animals are very old. So here's our wild type animals from this curve here, with this marked increase in total iron, and these are our long-lived mutants that do increase their iron content, but this increase is greatly lessened. And also shown here is a second mutation that's known to completely block this long lifespan increase. If we mutate a second gene called DAF16, this lifespan increase is completely suppressed all the way back down here to wild type. And if we look at these double mutants, DAF16, DAF2, this rapid increase in iron is restored and perhaps even accumulates to higher levels. Now, we published this study a number of years ago, and it gave us great insight into what specific process we may want to target to improve aging. Instead of thinking about genetic interventions, which clearly start at the earliest point of development, and are also known to have numerous other effects, particularly effects on growth and development, we started thinking, can we target iron in adulthood and later life? It's important to note that biological metals like iron don't just exist as free ions floating around inside our cells. They're nearly always complexed with other cellular components and molecules like proteins. To understand how and what iron-protein complexes change over lifespan, we next used a combination of techniques: chromatography, mass spectrometry, and proteomics. And from this, we learned that a protein ferritin, a highly conserved protein found in humans and in C. elegans, is failing in its job to safely store iron. So even though these animals, as they age and have nearly twice as much iron, the amount of iron being stored correctly and safely in ferritin is decreasing. The rest has somehow escaped and is likely damaging these animal cells, proteins, and DNA. So we think the same process is likely to be occurring in people as they age. So this left us with two new approaches to slow aging: reduce the amount of accumulated or excess iron in these animals once they have reached adulthood. And secondly, use a chemical inhibitor of lipid peroxidation, and this is known to inhibit ferroptosis. For the first approach, we use this compound, which I'll call SIH. It's a small lipophilic protein that's able to remove excess iron from cells and keep it from harming other proteins. For the second, we used a molecule able to stop the propagation of lipid peroxidation, or lipid damage. We'll call this LIP1 for liproxtatin. Initially, we checked the effect of these interventions on iron levels. Shown here are representative X-ray fluorescence images, again, where we're measuring the amount of iron. The amount of iron is quantitated here on this plot. Untreated animals accumulate iron as they age, moving from here to these orange points. Our iron-removing agent SIH significantly reduced this iron accumulation, shown here. And in contrast, our ferroptosis inhibitor, LIP1, did not alter iron, but we didn't expect it to. So next, what did these compounds do to the health of these animals? For this, we measured the maximum velocity these animals could move, measured per individual animal. Now, maximum velocity of movement is known to sharply decline with age and is an excellent predictor of both health span and lifespan. Seen here are untreated control animals at the first day of adulthood, and they're able to move with a peak velocity of 0.5 millimeter a second. At the age of median lifespan, at day 8, untreated animals have dramatically lost the ability to move. Treated animals also show an age-related loss of speed, shown by the blue and green data sets, but this loss has been significantly lessened, consistent with these treatments providing improved health in late life. We can see here. Well, what about the effects on lifespan itself? Both treatments, when given from adulthood after development, both significantly increase median lifespan. We can see that here. So the controlled untreated animals shown in orange, and the two treatments shown in blue and green. This is very robust data, and this data represents over 700 animals within each group. Lowering iron by SIH increased the median lifespan by 100%. And inhibiting ferroptosis increased median lifespan by 70%. And for context, the median lifespan for women in Australia is 84 years, and for men, 78 years. Data from the Australian Bureau of Statistics. Now, if we speculate here, if we could replicate these effects in humans, this would mean a shift in mean lifespan to over 130 years, with reduced late life frailty and perhaps reduced risk of disease. Note also the shape of these curves has changed. The maximum lifespan is only minimally affected, consistent with an overall effect of improving health span in mid and late life. Since we're targeting a cell death signal, ferroptosis, we next look for evidence of cell death in these animals in late life. We used a fluorescent dye that enters the nuclei of dead or dying cells in animals that are otherwise alive, albeit with increasing frailty. That's what we can see here. Within untreated animals, 20% of these animals at the median lifespan point had evidence of dead or dying cells, particularly within the intestine or nerve ring, the brain analogue of these animals. Lowering excess iron by SIH treatment or inhibiting ferroptosis by LIP1 both effectively blocked cell death at this time point. So in summary, targeting excess iron and inhibiting ferroptosis can significantly increase lifespan. Targeting excess iron in late life can affect health span with minimal trade-off, fitness trade-offs. And in limiting ferroptosis in adult animals may also promote healthy aging. So are these ideas ready for clinical trials? Well, yes, my colleagues Ashley Bush and Scott Aiton at the Florey are currently running a clinical trial looking at repurposing an iron-removing drug called deferoprone to reduce the progression of dementia. And we all eagerly await these results. But where next for my group? Well, SIH and LIP1 have been great proof of concept compounds, but neither are really suitable for further clinical development. Instead, we have started exploring new compounds with better drug-like characteristics. It's early days, and I apologise for not naming these compounds, but we've already identified one leading compound that looks just as effective, shown here by this purple curve on this survival graph, with the control population shown in red. It's reporting just over 100% increase in median lifespan. And this blue curve is an FDA-approved drug. We're modifying it, with the purple being our lead compound that deserves a closer look. And to that end, we'll next investigate the effects of these compounds on models of neurodegeneration and for possible benefits on health span. So we don't yet have a magic bullet to prevent aging, but we can all take steps to age as well as we can. The science still supports the advice that we've been given for a number of years. With respect to iron, it is important to understand that it remains essential for good health throughout life. In cases of iron overload, reducing these levels is important, but not to the point of anemia, which would create its own problems. So a healthy diet, eating whole foods rather than highly processed foods, remains the best advice, as is limiting sugar, salt, and alcohol. We should all maintain an high overall physical and social activity, as well as regularly exercising our minds. I hope you've had a chance to do that today by attending today's seminar. And with that, I come to the end of my presentation. And so I'd like to acknowledge my team and collaborators, in particular Nicole Jenkins and Ashley Bush, as well as my funding sources for this research, the Australian Research Council and Human Frontiers of Science.